जी, जी, Mrs. Nwachukwu, you are the host. Allow the lecturer in. He's trying to join. He couldn't join because you are not allowing him in. Mrs. Nwachukwu, are you there? Hello? Can you hear my voice?
Maybe you should call our line.
I don't have her contact too. Because she's not allowing the lecturer in. Although I've called Mr. Fasasi. Okay. Mr. Fasasi, please. Allow doctor in. God.
Don't worry. Let me just. I can't be the host again. She's the one that can do that. Let me see.
He's not even
Okay,
online.
I'm the host. I'm the host. Don't worry. I will invite him. Don't worry. Maybe I need to invite him from here. That's just it. But normally, it should. The link should work for him. I don't know why it's not. I just felt people can't do.
On the system he's using. So
I think it's his network. Maybe his network. I really be his network. Shouldn't have any issue.
Okay.
No problem. Let me just try and invite him again. Okay. Settings. Posters. There's no post. There's no lock. Why it's not going on?
Àwọn aláàgó mẹ́sàn-án ìtọ̀ yìí tí táyà.
I am sending him a new link, so let's see how that goes.
Okay, does it mean we have to leave this one?
No. It is the same link, but I'm copying the link directly from the page. So
Okay.
I've sent it to you, Ms. Akinleye. Ms. Akinleye, you can share that with him.
Okay.
Let's see that. Once that, if that does not, that means it's his device. He needs to change the device. Well, the meeting is still on. Let me go to here. Control.
I've sent it to him.
Is he responding to your chat?
Yes, we have been
So that,
communicating.
okay, so that he can just click
Let
on
me
it.
let me put a call to him again.
Okay.
Mr. Fasasi, he said he's
Yes,
trying to connect. That is connecting. It's just rolling, and he's
that
using
will
two
be
devices. He's
that will be his
using
network.
device. The two
It will be
devices
his
are not
network.
doing.
That's the network. If it's rolling, other people can join now. I've just
Yeah.
joined back, so it's his network. That's beyond me.
Okay.
That's
Let's
beyond
wait.
me.
Let's wait and see.
Okay. You want me to end this and create another link? What do you feel? if it's not joining.
Will it be able to enter without new link?
No, that means we will create a new link for everybody. I will end this, create a new link for everybody. He join early, then every other person join after him.
Let's hear other people's opinion if they're okay with that.
I'm okay with that.
That will be fine. You can create another link. Let everybody join.
Okay.
Somebody has joined. No? Let me see.
So we should leave this one.
Is he?
Are you
Hold on.
If
Okay.
he's not able to join, maybe we just close this, create a new link. More and more people are leaving and joining. So what do you say? Because of the time, so let's make the decision quick. I have some other things I need to attend to.
Hello, good evening. Oh.
Hello?
Finally.
Oga, welcome.
Welcome, Zao.
Welcome,
I've been trying to connect since.
I think it's the network, sir.
Is a network then?
Yes, network.
Can you see my slide?
Yes, sir.
Oh, please. Let's take systemic fungi, please. Because that was what we wanted to do like last time, before it goes off. So, are we ready? Can I continue?
Yes, sir.
Yes, sir.
Good evening.
Very
Good
much
evening,
ready,
ladies.
sir.
Yes, good evening, ladies and gentlemen. So we've discussed general mycosis, all right? And we stopped at diagnosis or something. Who can remind us what we did? Brief. Can somebody remind us?
Yes, sir.
Briefly, please.
We talked about the general characteristic of fungi.
Okay. Yeah, okay.
Then you discussed about the types of mycosis.
Okay.
Then we were able to talk about the opportunistic fungi.
Fungi, okay.
Which includes the candida,
okay,
and other types of fungi that you were able to explain that there are some conditions that trigger candida, which includes antibiotic use, immunosuppression,
immunosuppression.
Yes, diabetes, and other conditions, poor conditions.
Thank you. We discussed about antifungal drugs. All right, we briefly discussed antifungal drugs. So this morning, this evening, we talk about systemic fungi. All right, which is also called systemic mycosis. All right, when they say systemic, it infects the blood, and most of these fungi, they infect people during inhalation. By inhalation, sorry. All right. When people inhale the spore of these fungi, they develop the lung and they escape to the blood, and they infect the bones, the livers, and all the sensitive organs, include the skins. All right. So the common among them is to plasmosis. Others are blastomycosis, coccidioidomycosis, and paracoccidioidomycosis. So these are systemic infections. So as I've said, is a respiratory tract infection, and the symptoms usually by coughing, like that of tuberculosis, then fever, because it affects the blood. And ideally, when you want to diagnose this kind of infection, you use blood culture, all right? If it's in the blood or if it's the lung, you use sputum, all right? And the antifungal treatment, which can be used to treat these fungi. And I want to say this. The problem is exacerbated by low immunity. When somebody has a condition that makes the immunity drops, like when you have HIV or you are on drugs that suppress the immune system. All right. And most of the fungi that cause this infection are dimorphic. Well, I think we discussed dimorphic this other time, whereby at room temperature, they are like yeast. At room temperature, they are like mold. All right. And at room temp at body temperature, they are like yeast. All right. They are dimorphic. So if you want to diagnose this infection, you have to incubate them at room temperature as well as at 37 degrees Celsius. That's 22 degrees Celsius and 37 degrees Celsius. So first example, we talk about cryptococcus, and the common one is neoformans, which we all know, all right? And it's caused by when you are exposed to infected bed droppings, like pigeon, all right, bats, all right? And if, unfortunately, the immune system is compromised and weakened, such as individual with HIV and AIDS, or somebody with organ plant who has had organ plant exchange. That person, because they be on immunosuppressive drugs, so and the problem is further marked. So what are the kind of symptoms you have? Because it's a organism that gets to man through the lung during inhalation. So the common symptoms are cough, chest pain, and fever. And in most cases, if it spreads to the blood, it enters the brain and spinal cord, and it causes cryptococcal meningitis. So and you start to have symptoms like headache, fever, convulsion, not convulsion, sorry. I will change it. Neck pain, nausea, and sensitivity to light, which are the signs of meningitis. They are symptoms of meningitis. So it can infect the skin and bone and other organs, such as the liver and the kidney, all right? And in such case, cause severe cases, especially in people that have severely weak immunity. How do you diagnose the infection? You know, in most cases, as I told you, is an infection of the blood and spinal cord. So what you use is blood. In most cases, meningitis. So use spinal tap. All right. Then you can do culture, microscopic culture, and antigen test. We will discuss all these things, laboratory diagnosis of fungi later. So how do you treat this infection? You know, the infection is if it occurs in the lung, if it's detected in the lung, you use fluconazole. But if it's spread to the meninges, you have to use amphotericin B with flucytosine, and followed by many months of fluconazole antifungi. All right. So cryptococcal is a common cause of illness and death in people with weak immunity, as I've said. So it has to be treated immediately and with appropriate antifungal therapy. So let's discuss I want to say this: all these systemic infection, they are inhaled and they cause infection of the lung and before it spreads to the blood. So blastomycosis, all right, it's caused by inhalation of dust, especially in an area where we have moist soil or decaying organic materials, such as roots and leaves. As I've discussed, the spore can spread into the air and it's inhaled by a victim. As is inhaled, the infection starts in the lung and spread to other parts of the body, such as the skin, bones, and brain. About half of the people develop sign of illness, which is usually pneumonia type, such as cough, fever, night sweat, muscle joint pain, weight loss, and chest pain. How do you diagnose and treat the infection? Is by collecting body fluids such as sputum or biopsy of the tissue. And you can culture it in the lab and do microscopy. You can see the yeast-like budding under the microscope. Treatment is by use of itraconazole and amphotericin B, followed by use of about 6 to 12 months of azole antifungal medication. We all know that antibiotics that works for bacteria is useless against fungi. So it will not affect blastomycosis and should be treated immediately to avoid complication. And lucky enough, the blastomycosis is not contagious, but it's limited to people of certain regions, especially people that have weakened immune system. They have high risk of severe diseases. Now, the next one is histoplasmosis. It's caused by Histoplasma capsulatum. It's also found in soil, and it can also be got from droppings of the birds and bats, like Cryptococcus. So it's inhaled like all the spore is inhaled like all the other systemic fungi, and this one is common in North and Central America, but can also occur worldwide. So nobody is safe. So the usual symptom is often mild, sometimes it's symptomless. And since it affects the lung, you have a flu-like symptom, such as fever, cough, and chest pain, chills and headache with fatigue. If the infection is heavy or the person has low immunity, the infection can be spread to the organ, and it can affect the liver and the spleen, causing spleen enlargement. It can cause mouth ulcers, and the infection can be very serious. And in most cases, the infection resembles tuberculosis with weight loss and night sweat. So one has to be careful so that you don't use the wrong drug. So the diagnosis can be done with fungal antigen testing, chest X-ray and CT scan, and in the laboratory we can culture the fungus from sputum, blood, and tissue samples. So let's go to the next fungi, which is coccidioidomycosis. It's often called valley fever, and the organisms that causes it are Coccidioidosis immitis and Coccidioides passadazole. As I've said with others, they live in dry soils, often found in southwestern US, including Mexico. Sometimes they are found in part of Central and South America. And as I've discussed with you, the infections by inhalation of the fungus spore, and they can be healed, and it's found in construction area or agricultural area where there are a lot of activities and there is soil disturbance. So as in others, you have flu-like symptoms such as fever, cough, and chest pain, fatigue, headache, joint aches, and sometimes rash. But let's say sometimes the infection can be mild or asymptomatic, and it can affect about 1/3 of the population. But it's very devastating in people with weakened immune system, as we have discussed. And it can cause a long problem, even life-threatening, that is, if untreated, and it can kill. That's what it means. The diagnosis is about, as we have discussed, in the laboratory, you need blood tests, antigen testing. You can culture the specimen and you can identify the fungus in the laboratory. And X-ray and CT scan can help in clinical diagnosis. So what are the antibiotics? You can use conazole or itraconazole. And if it's systemic or it's very single therapy with using amphotericin B. And let's know this, there is no vaccine for coccidioidomycosis. And prevention is the main focus in minimizing. So those are the systemic fungi we've discussed. So we are going to cutaneous mycosis. And these are, when we say cutaneous, these are fungi that affect the skin, the hair, and the nails. And among them, 1, dermatophyte, 2, yeast, and superficial fungi. Those are the one that, and we treat them one by one. For example, dermatophyte, that is Trichophyton, Microsporum, and Epidermophyton. They are often called tinea, and they affect the body. When they affect the body, they are called tinea corporis, and the common one is ringworm. Ringworm. And when they affect the scalp, they are called tinea capitis. When they affect the foot, causing athletic foot, they are called tinea pedis. When they affect the growing of jock and they cause jochitis, they are called tinea cruris. And when they affect the nails, they are called tinea unguium. And Candida can also cause cutaneous mycosis. And as I told you, as we have discussed, in people that are obese, that have skin folds that traps sweat, is very common. So it affects the skin fold in moist area. And another one is Malassea species. It causes cutaneous mycosis too. Example of Malassea is Pityriasis versicolor, which appears discolor patches on the chest, back, or neck, especially in young adults in hot or humid climates. So let's go to dermatophytes. We are treating each one by one. Dermatophytes, also known as tinea or ringworm, they are referred to a group of fungal infections that affect the skin, the hair, and the nails. All right, as we have said before, they are caused by dermatophyte molds, and they survive by digesting and grow on keratin, not keratin, keratin. That's the protein of the skin, hair, and nail. The main generally that are responsible are Trichophyton, Microsporum, and Epidermophyton. So dermatophyte is clear body area. If it's in the body, if it's found in the body, they are called tinea corporis, and they cause red, itchy, circular rashes, often classified as ringworm because of the shape. If it's athletic foot, they are called tinea pedis. They cause scaling, itching, and featuring between toes. If it's on the scalp, they are called tinea capitis. They cause scaly patches with hair loss, usually in ring form. If it's the growing, they call jochitis, and this is tinea cruris. So it causes raised red lesion with central clearing. If it's on the nail, this is called onychomycosis, caused by tinea unguium, which causes thickening and decoration of the nail. And with this, the infection is spread by contact, direct contact with fed people, or with animal or contaminated surfaces, such as floors, toys, and grooming item. That's why I say if this infection are infection of poverty or poor hygiene. So what are the method of diagnosis? In the laboratory, we use microscopy method. In fact, in most cases, dermatophytes can be identified microscopically. You can also culture it, and they have a characteristic visuals. And they are found globally. And in most cases, tinea capitis affect frequently the children, that is tinea on the head affect the children. Tinea cruris, which affect the growing, are found in elderly. So let's discuss superficial mycosis. So these are fungi that affect the outermost layer of the skin, the hair, and the nails. And they will not invade the living tissue. The skin, that's the dead part, they affect the dead part of the skin. And they are mainly cosmetic, and they are mainly cosmetic, and they really cause serious health problem. So they usually cause minimal or no inflammation responsive from host. Example of these ones are Pityriasis fascicolor, often caused by Malassea species. They present as discolored scaly patches, usually on the chest, or back, or neck. There's another one, Tinea nigra, caused by Ote wanenki. They manifest as brown or black patches, typically on the palm or soles. There are pedra. There are two types of pedra. The black pedra is caused by Pedra ote, which shows hard black nodules on scalp hair. White pedra is caused by Trichosporum species, which is identified by soft white nodules on facial, beard, or pubic hair. What are the features of superficial mycosis? They do not cause, or they do not invade deeper skin layer, as I've said. They do not invade the internal organ, and they do not provoke significant discomfort. Diagnosis is by microscopy of skin scraping or hair samples. And treatment is topical, and rarely oral antifungals are used. And as we have said, they are mild infection, and they cause cosmetic concern. But effective hygiene antifungal therapy can prevent reoccurrence or spread. So health is wealth. Personal hygiene is very important in preventing mycosis. Now, let's discuss general property of fungi. That's the next part. Are you with me in the class? Oh,
Yes, sir.
hello.
We are with you.
Okay.
Yes, sir. Yes, sir.
Okay. Let's discuss general properties of fungi. We all know fungi are multicellular, mesocellular eukaryotic organism characterized by unique structure, physiology, and ecological property. They are distinguished from plants and animals or bacteria. What is the cellular structures of fungi? They are membrane-bound nucleus containing DNA wrapped around histone protein. That is, the nucleus is membrane-bound, all right? And the nucleus contains DNA, and it's surrounded by histone protein. And the cell possesses mitochondria, Golgi bodies, and the plastic reticulus similar to plants and animal cells. The cell wall has a defining feature containing many chitins, glucan, giving strength and rigidity, unlike plant cell wall, which are primary cellulose. So they have chitin like insect, and glucans like human cells. The plasma membrane is stabilized by ergosterol rather than cholesterol as in animal. So the plasma membrane has ergosterol instead of cholesterol as in found in animals. What are the morphologies of fungi? How do they show features, observable features? The most fungi grow elongated thread-like structure called hyphae, and a network of hyphae form mycelium, which is the main body of the fungi. All right, these are mostly multi. All right, they form hyphae, which are elongated threads, and when there are many of them, they are called mycelium. Some fungi are unicellular. They exist as yeast. We all know example of that, and they reproduce by budding of fission and can switch between yeast or filamented form. So in dimorphic fungi, as we have said, you've said it many times, at one temperature, which is at 37, they are yeast. At room temperature, they are filamentous form. So they can switch from one form to the, that's dimorphic fungi. This hyphae we've discussed, which is thread-like structure, can be divided into compartments by septa, which contains pore for cytomeric streaming and organelle movement. Some hyphae are segmented with septa, some are not. And as we have said, many fungi produce spore for reproduction, which can be sexual or asexual. So they reproduce by production of spore. And the spore can be sexually produced or asexually produced. Nutrition and ecology. Fungi are heterotrophic, that they can absorb organic matter from the surrounding by secreting enzymes to break down complex substances. And because of this, they are the major decomposing organism in the generation of matters in nature. They can be saprophytic, that is feeding on decaying matter. They can be parasitic, causing disease in plant, animal, or human, or mutualistic, forming symbiotic relationships, such as an example is mycorrhiza with plant roots. All right. And let's note this: fungi lack chlorophyll as found in plant and cannot photosynthesize. That's very, very important. They look like plant, they are not plant. They look like, sometimes they look like animal, they are not animal. Ecological and functional role. Fungi are essential decomposers in ecosystem, recycling nutrient and organic material. Some produce metabolite like antibiotics. We all know penicillin is produced from fungi, which other have roles in food production. We know that we use fungi to produce some cheese, and they are very important about technology. So are we okay with that? Can we go to pathogenesis of mycosis? Can we go on? Class, can we continue? Hello? Hello?
We can, sir. Yes.
Okay.
Yes.
Yes, sir.
So let's go to pathogenesis of mycosis. Pathogenesis of mycosis is a problem which fungi causes disease. How do fungi cause disease? And when you want to cause disease, you have to have a means of entering human or your victim. You have to adapt to the victim environment, and by adaptation, you multiply so that you increase your number and size, and you be able to invade the host defense. And when you invade the host defense, then you can establish infection. Those are the mechanism of pathogenesis of most organisms, and mycosis fungi can achieve all this. So let's start with mechanism of fungal pathogenesis. Let's start with entry of and colonization. Fungi typically enter body via inhalation, as we have seen. Most of the serious infection of fungi are through spore inhalation. And in most cases, it can also infect now by direct skin contact, or when there is breakage on the skin of the skin surface due to wound, they enter. And once they are inside, they are there to the host cells. By resisting physical removal, it's very difficult to remove. And with this, they compete for nutrients in host tissue. Virulence factor. Fungi produce enzymes such as protease. Please, no, please, who is talking? Oh, fungi produce enzymes such as proteases that break down protein and phospholipases, which are used to degrade host barrier, allow invasion and tissue destruction. Some fungi secrete toxins and immunomodulatory molecules that damage host cells and interfere with normal immune response. Invasion of host immunity. Fungi have adapted multiple strategies to invade immune system, include one, surface masking. Special cell wall components, e.g. melanin and glucans, hydrophobin, mannans, and capsules, hide fungal antigen from immune recognition. Morphological changes too. The morphology can switch from yeast to mold to better survive or evade the host immunity. Formation of biofilm and intracellular survival niches help some fungi and to endure hostile immune environment. Immune response and diseases. Successful fungi trigger weak or effective immune responses, leading to persistent colonization or systemic spread in immunocompromised patients. Fungi may proliferate unchecked, causing severe invasive diseases. So, key steps in pathogenesis of fungi: adhering to host cells, invasion and tissue penetration, nutrient acquisition and growth, resistance and modulation of host immune defenses, spread and persistent in host tissue. So, they either cause death or cure with antifungal. If you are infected with fungi, you cannot remove it physically. Can we stop here today? I don't want to overload you. I know you have been having lecture. So that we'll continue laboratory diagnosis of fungi diseases next week. Are we okay?
Sir, I think we should
Hello?
continue.
Yes, sir.
We should continue?
Yes, sir. We're okay.
You
Yes.
are not
No,
stressed?
sir.
We have
No,
been
we are
two lectures.
It's okay. Let's start with laboratory. Let's start with laboratory diagnosis of fungal infection. So this for laboratory, what are the specimens we require in the laboratory, especially for dermatophytes and cutaneous, especially cutaneous fungi? We need skin, ear, and nail sample. So the first thing we discuss is skin scraping. And usually before you collect your sample, you must clean the surface with 70% alcohol. And you can use disposable scalpel blade for scraping of the soft inflamed area. All right. You can also use you can also use, if you don't use scraping, you can use strippings. This is an alternative method for collection of skin specimen. This method is to apply waterproof transparent vinyl adhesive tape, 3M type CZ1, firmly to the affected area and peel it off. So you apply the tape, that's the cellophane tape, to this infected area, then you peel it off. And while you are peeling it, you know that the skin layer will contain some fungi. Then you apply it to a sterile glass microscope slide. This can be transported to the laboratory. This is often used for isolation of dermatophyte and yeast by culture. This method is not suitable for microscopy examination, except in case of Pityriasis fasciculum. That's Malassea species. Nail specimen. Friable material should be removed from under the nail, or clipping may be taken with scissors or nail clipper. Scraping should be taken with scalpel blade. Where yeast is suspected, exudate is expressed by probing with a flat excavator and collecting on a mostly sterile swab. So if it's skin scraping, you can use scissors, you can use clipping, you can use nail clipper. All right. But if your specimen is moist and you are suspecting yeast, you use sterile swab. If your specimen is air, it should be removed by plucking with epilating forceps and never by cutting. You understand that one? You should remove it by plucking, like if you are removing from the root. The reason is that most of the fungi are at the root of the ear. So if you cut it, you may not be able to get a best specimen. So you can also use woodlamp, especially in ringworm affected ear, which allow the infected area to flourish for better selection of material for examination. How do you store and transport skin, nail, and ear sample? These are keratinous samples. They contain keratinous material, and you should allow them to dry out to prevent the overgrowth of saprophytic bacteria and fungi that occur if moisture is retained. So what you use is black paper, which is folded to a form of a pocket, like an envelope, or envelope can also, special envelope can also be used for collection. The common specimen we use this one is ringworm fungi, and they can remain viable for weeks or even months. Note, your specimen for ringworm examination should not be refrigerated, and you cannot use swab. Swab is only used for yeast infection, as I have discussed before. For sputum specimens, the first organism you should think of is Aspergillus organism. And they can be very in number, size, and they can overgrow. So what you usually use for sputum is to collect 3 days specimen collection, and we should batch it 3 hourly. And sputum should be refrigerated to reduce growth of saprophytic bacteria and yeast. All right. So that's as far as sputum collection is concerned. For cerebrospinal fluid, lumbar puncture of minimum of 5 minutes is recommended for cryptococcus in non-AH patient. That is, normal patient, you can collect 5 ml. And this is transported to the laboratory without delay. So it has to get to the laboratory immediately. Still warm. All right. So how do we diagnose yeast in the laboratory? Direct examination. So by the microscopic examination, give early indication of the presence of yeast. So you can observe capsules surrounding yeast cells in CSF provided by presumptive diagnosis of cryptococcus. All right. So once you examine yeast cells and you are seeing capsule, you should know that you have cryptococcus species at hand. Isolation of yeast. Culture media. The culture media we use for yeast is, as I have said last week, is Sabro medium. And this one is used worldwide. You can also use moth peptone agar. All right. All pathogenic yeast grow at 37 degrees Celsius. All right. And you can have a very good growth when you incubate for 48 hours. How do you identify yeast? On the culture media, which is Sabro, they appear as pasty opaque and usually pale color. The color is white or off-white or beige coloring, over with sweet reminiscence of ripe apples. And you can confirm microscopically by emulsifying a portion in distilled water or in sterile saline. Or this is what I want to, you can emulsify lactophenol cotton blue. I wanted to remember this last week, I forgot. lactophenol. This is a common stain we use in the identification of fungi. You can also emulsify in iodine, and we will observe the characteristic budding cells in yeast. You can also use jam tooth test. About 90% of yeast isolated from human material are Candida albicans, and a rapid method of identification is to use jam tooth test. This is due to the ability of the species to produce pseudo jam tube in serum. And for the jam tube test, light touch or single colony with loop of pasture prepare. You use it to remove, you remove excess inoculum and emulsify the cell in 0.5 ml of all serum or other serum in a small test tube with a loose cotton wool plug. You incubate at 37 degrees Celsius in water bath for 2 to 4 hours. Don't let it be more than 2 to 4 hours. I did is to incubate for 2 hours. And the pseudo hyphae can be seen. And example is shown on the pictures on the screen. You can see yeast cell with budding, yeast with pseudo hyphae, and the other one with hyphae. There's another one budding these cells A and B. So these are, I think we should stop here tonight, my people. Let's go and rest. We'll continue next week. Are you with me?
Yes,
Hello,
sir.
everybody. You have been having lectures since
Thank
5
you,
o'clock. Thank you very much.
Well done.
So, well
Thank
done. So we
How
continue
do
with
we
syllabus.
get our notes?
I'm showing you your notes. I'm going to give you a slide like this. This is a slide that we just make sure that I'll just make sure it's good for your revision. That's all. It's for your revision. So let me finish what we are doing. I think by next week or upper week, you should be able to finish everything, and I will send the note to the wall. That's the WhatsApp wall. All right. Is it okay with you?
Okay, sir.
Yes, sir.
Thank you, sir.
I will send it to you.
It's
I will
okay,
make it
sir.
more comprehensive. Okay, I will make it a bit comprehensive. But whatever the lecture is to provide with basic facts. You can, if you have time, you can read more. But what I gave is enough for you to pass your exam. I wish you all the best. Have a nice night. Good night, everybody.
Thank you, sir.
Good
Thanks,
night,
sir.
sir.
Good night, sir.
Good night,
Good night,
madam.
Ogami.
Yes,
Hmm.
yes, sir.